Just to get the ball rolling, right? So with regard to 4, even though I'm also a scribe, getting the what's needed for optimal deployment, and you touched on this, Ron, that it would be as part of the standardization. So if we do decide to go with antibody-based, which is the topic of this little breakout, we'd want to, for different methodologies, different tissues, we want to have panels and clones of validated antibodies, preferred buyers and so on. And that would be the first thing, like a list of clones, antibodies, where they're sold, and which tissues and which fixation, along with a standard or a couple of standard fixation protocols.
Any other statements on this? Because I'm going to start speaking, which we're not supposed to do at length as moderators, but if no one else contributes, there isn't a lot to discuss. And I think one of the problems that we have had in talking to other groups is that there are many antibodies out there, and not all of them actually really work, or at least are validated. And on human tissue, there are only a limited number of validation approaches that can be taken. And so one item I think would be very useful for the community to work on is a consortial approach to cross-validating and then listing in a public place the specific clones, even the conjugates that people have validated, how they're validated, and creating a much larger panel list that people can use for the multiplex approaches. Now that may vary for the ones that are chelate versus fluorescent. I'm leaving histochemistry per se out of this since that's not what most people are using. But I think that this is important to do. And it raises another question having to do with platforms, which is that there are some platforms that are open in the sense that you can pretty much choose any antibody and incorporate it into your method fairly easily. There are others that are closed, that are proprietary, that go with particular systems. And I'll make one mention. I'm not endorsing or not endorsing this technology, which isn't even rolled out yet for Miltenyi, but it absolutely depends on their proprietary release antibodies to work. So they have many of them, but it's a closed system. So do we want the HCA to come to any standards on that, or should everybody just use whatever works for them?
So Ron, this is Stephen Hewitt. I think you've stated this very well. I think that antibody specificity is something that, since most of the specificity assays are performed outside of the multiplex, should be relatively standardized. However, it appears that the different platforms are sufficiently different enough that the validations really become very platform specific. Now, I think it is reasonable to anticipate that many clones will cross-validate. And so you probably are kind of looking at a multi-tiered database where we have specificity agreed on, and then validation data shared across platforms so that people can understand which ones are working where and where they may actually give us a common overlap across platforms.
Perfect comment, absolutely. I think that we should compile a list of the different platforms and then begin to do the lists and the validation standards according to those platforms, and hopefully multiple groups on any given platform, so there's again some cross-validation between different laboratories to ensure that things hold up. And so that's really one action item, is to get together a group that represents each of the major technologies, and then
Hi, it's
consortial action to do the. Elizabeth?
Sorry, my thing got laggy and I did not mean to interrupt anyone. So I know HubMap, we have a list of our antibodies, and I know we're specifically talking about HCA now, but would it be worth for all of the different NIH consortia to come together and share the antibody lists? Because it would be probably really useful to use similar antibodies when possible. I don't know if my voice got through all the lag, so sorry if it didn't.
Yeah, it's pretty clear, and that's a very good point, that why duplicate efforts in siloed ways as opposed to joining everything together in one larger list. So in the action, we should expand from what I just stated to other appropriate groups that are building out large lists for multiplex through other consortia. And Richard, maybe you can really help us, and Elizabeth and others navigate that environment.
Yeah, I can go ahead and share the HubMap one, if that would be helpful. But I'd be happy to help with an an outer consortia conversation with antibodies, because I think it would be nice long term.
So let me just add a little bit to this. So we started this within HubMap, taking a sort of multi-step approach. So the first was to gather together the list of antibodies, and that's the process we've gone through. And the second step is the much harder one, which I think is really getting to understanding how the antibodies have been validated, both internally within the consortium as well as externally by the vendors. So I think for me, there's an issue there in how we think about reducing down and describing how antibodies are validated. So I think that's one issue. And a little bit of what Elizabeth was just mentioning, there are other consortia at NIH, and I'll pick on ENCODE as the example here because Dan's in the group. So they already have their whole list of antibodies that they use as part of ENCODE up in their website. We've tried as much as possible to follow the same format, thinking that at some level we do want to try to make use of the data that ENCODE's already put out to understand how what we have done relates to them. But again, it seems like for each different program, there's variations in how they have, first of all, put together their list, and then secondly, in how they think about validation, and then downstream, what does that validation mean? For example, do they use that for some kind of quality score on the data that's being produced? So, it seems like there's the reason for doing this is really understanding the quality of the data that gets generated downstream, so that people have an understanding of how that data was generated using which antibodies. So it seems like for the transition between each different step here, there's a new set of problems to address. And I think that's why part of what we were hoping to get out of this discussion is, given that several groups have already started building lists and some groups have different approaches for how to characterize the validation in their settings, is there a more generalized way of doing this? And, for example, Emma, through the Human Protein Atlas, has a lot of experience in this type of approach, given the breadth of what they've done. So, I mean, I think I'm interested in hearing some of where other people have got bogged down in trying to do some of this.
Hi, this is Emma. Sorry, I had to log out and log in, so I missed the beginning of what you were saying. Could you please repeat?
I'll take it, Richard. Emma, the question is, how is your group validating antibody lists or reagent lists?
Well, we spend a lot of time on validation, I would say most of the time. So we try to do what we call enhanced validation. So either independent antibodies that target different epitopes, either validate by that, validate by IP mass spec, so that we recognize that it's really pulling down the target protein, or by gene silencing or knockout, that we knock out the target protein and see that the signal disappears. Or we also validate with GFP tagging, which is a bit harder because we can basically show that the stain, it's easy if you have a very specific expression pattern that, for example, shows a temporal variation or something, then it's easy. But if you just have a nuclear protein, it's pretty hard. It can just be another nuclear protein, right? You don't know for sure that it's the same one just because you co-stain with antibodies. So that one, I would say we're doing it, but it's not a perfect one. Ideally, knockouts or paired antibodies or IPMS. And so far, I think we've done that for 25% of our antibodies, and we worked for a long time. So it's a lot of work.
Let me make a comment on that unfortunately, at least in our experience, you can knock something out in a cell or a cell line and get what looks like a clean signal, and you go into a tissue and it's not the same. And so we need to be thinking about standards that work in a tissue environment. So the minimal standard is that it's pretty clean on a cell where you knock it out. but that is probably not a sufficient standard. So I think the action item here for all of us as a follow on to the general part of sort of making the list is that we need to have, if they're not the same group, a group working on some agreed minimal standard of validation. So when somebody says that they really have done this large set and stain with X, Y, and Z, that people can buy that. And that also goes into a next part of the discussion, which is how do we actually share these data, and how do we run any kind of standards that let us do what you would do in the nucleic acid world in terms of cross-normalizing data sets in terms of intensity and patterns of staining. But let's finish up with the antibody part. I think we have sort of two action items and sort of general agreement to make use of all the different approaches people have been taking, the lists that are there, and to work within a group across platforms to create the best curated list that we can, and at the same time work on coming up with a very clear standards in an area where it's non-trivial to do all the controls we would like, to try to come up with some reasonable validation plan. Anything else on this topic, or maybe we'll go on to the next issue?
Hi, this is Emma again. Can I say another thing? Something that we find pretty useful is to screen with protein arrays or anything like that. Not that that an antibody works well in protein array, that it detects a target protein and not detect any other antigens. It's not proof that that antigen will ever work in any other assays, but but the antibodies that fail in a simple protein array, they tend to fail in most other assays too. So we use that as a first screening to remove antibodies that we don't waste our time on anymore. So that's also a pretty good tool to limit the number of antibodies that we actually think are useful.
Do you actually run your protein array through a fixative before you test the antibodies to see if you lose epitopes?
No, we don't. We've done that and it's not a big difference, but our antigens are not full-length proteins either, so they're most likely not folded to start with.
And don't have lysines in them. So.
Sorry?
Then presumably don't have lysines that get modified.
Well, they're producing bacteria, so there's no PDMs, but there might be lysines in them, yes.
So I'll jump in on that one and say that we've done similar studies, and in antibody validations, the group that Emma worked with and a group that I validate the antibodies with collaborate extensively. I work with the NCI CP Tech data set for antibody validation, and we do see some differences in performance between native protein lysates versus cross-linked specimens. Oftentimes this has to do with substantial differences in antibody evolution rather than necessarily substantial changes in specificity.
Okay, one other subtopic on this subtopic has to do with the need for new antibodies. You know, we have, and I've talked to other groups, have run into problems that there are clearly determinants, targets that folks would really like to get at. They know they're there. There are not very good reagents for it. Do we need to come up with some plan, or at least some recommendation to whoever the right agencies are about supporting an effort to expand the panels? And maybe Richard or some of the other folks that are involved.
Well, I wanted to ask you a quick question about this, and this is probably more from my ignorance than anything else. So is one of the challenges of developing antibodies, particularly for targets that people have been interested in, the challenge that the target itself, the protein itself, is quite variable? I mean, how much of a challenge is this for at least what are quite often described as kind of high value targets that people would like antibodies for, but which we don't seem to have successfully generated good quality antibodies for yet? So, it seems to me there's, at a very basic level, there's two challenges. One is the variance in the antibodies themselves, or whatever you're using in terms of the affinity region for a particular protein target. And then there's the variants in the protein target itself. And it gets a little bit back to the discussion that just happened around whether it's changes in the nature of the protein target from tissue to tissue, so intrinsic properties of that, or the way in which the tissue itself has been processed, whether it's things like post-translational modifications. How much of all of those changes make a difference in being able to generate new antibodies?
So I'll try to make a couple quick comments on that. I think that for diverse targets, we actually do well. Where we run into substantial problems are highly conserved protein families. Hox genes are a great example. Sometimes inferior genes are another example. Obviously, developmentally regulated families of genes, oftentimes filled with transition factors, remain a number of good specific antibody reagents. Large polymer proteins, things in the intermediate family, collagen stuff, can be difficult as well. And again, it has to do with homology of amino acid sequence. The other issue that we're facing, especially in multiplex assays, is that although we can get a few polyclonals to work, it's not like immunohistochemistry where polyclonals are still very useful. In most of the multiplex assays that we've evaluated and seen data on, you really are forced to highly purify clones. And if the purification features of those mono sometimes seem to be the feature. I don't have as much experience with this as the people on the phone, but I've heard report that you have to have monoclonals in serum-free media effective in all instances. And this makes sense, but all because of the challenge how is antibody to purify them.
All right, we have 15 minutes left, so I'm going to move on from this particular topic and want to try to cover one or two other things. We haven't talked about standards. We talked about validation, which is a type of standard, but in terms of the actual data sets that emerge, what do we need to do in providing the kinds of data that people can then use to try to interpret themselves what is really in those images? Should people be putting up all the original TIFF files? Are we talking about process data? How do we know that that processing has not obfuscated or changed things? Is there any way for people to be able to do data reevaluation on any of these kinds of high-plex samples? So where are we in that regard?
So the AN is just to release data for it. And one of the ideas was to link also data process pipelines that were included, so that you would have like a repository link to a very specific computational pipeline and the metadata and all the information for the instrument. So you could theoretically go back to the raw data and just add as a file, as a YAML file, and then process it with this parameter. So I think this is something that we're definitely looking at imaging group to really make this more reproducible, and also decide what really goes into an OME. We decided as like one of the operating process.
I heard some of that, Denis, but you'll deal with the scribes, and we'll get that into the little doc at some point. Anybody else on this issue of standards for data reporting and potential reuse?
Hi, Ron. Hey. Good. Let me just chime in. I have I came in 4 minutes late, but I love the discussion. I just wanted to know things. Number one, another follow-up on a topic that so we can continue the arc of this conversation. I'm taking notes. I think it's a framework. Issues come up over and over again. Not garbled like last speakers. So audio tomorrow well. So I'm going to create a spider diagram that just brings up the 7 or 8 different variables that are always in different ways. Try to get a conceptual framework, talk about this and how to invest in different areas and knocking down all these challenges. And then I just want to come back to Stephen Hewitt's point about the multi-families and the hawk, all of the issues. It is possible now on what isoforms are pulled by an antibody and mapping. So this is a very active area within HubMap. So I'll stop there, but thank you.
Jonathan? Is there anybody else with comments about this kind of standardization?
Yeah, it was about reusing. Are we really reusing images? You sort of had a bottom line question like antibody-based spatial imaging. Can we reuse images? Is there concordance? How do we measure concordance across images, et cetera?
If I caught what was just said, can we reuse things across images, reuse data? One thing that is a much bigger problem here, or a big problem in general for the HCA, and a big problem also in tissue, is that it's very hard to put a standard on every image data set so that it can be used for cross comparison. So I'm not sure that's a direction we can actually go, but we should give some thought to whether there's any kind of tissue that could be obtained that would be usable for at least some kind of cross normalization across the data sets.
Hi, this is Anne. I also want to say something we do, for example, and things like work well from antibodies and it's for antibodies. And what we found when we discussed with collaborators is often that we get the same result. Some differently hard. So sharing lists of reagents have been successfully conjugated versus not successfully conjugated, I could feel a bit to avoid that we're all doing the same. That's a pretty simple sharing. I can for pancreas project in the EU example started such a in general an image repository that allows and we interpretation of image that are not, but all the raw data would be amazing as well.
I would like to end in our last 2 minutes on, even though it's a little bit off the way we're supposed to do Crocs Up, on an aspirational issue, which is, does it matter how many plex we aim for? This is a debate between the optimal ways of doing single cell sequencing versus more cells to a shallower depth. And the question is, does it matter how far up we push the plexity of our imaging data? Does it enable us to generate more hypotheses by being a little less constrained and having too much more in what I call black space? So it'd be interesting to know what people think is the right range to aim at, where we could then roll something out to be used fairly widely across the consortium. Any comments on that point?
I would comment, Ron, that the plex maybe changing question in like how many cells. Maybe for certain organs, we agree upon the key cells that we want to be able to phenotype and at which depth, right? So maybe that would be the standard, like markers that are shared, and that's why the common panels and clones, but markers that are shared to phenotype distinct cells across the different organs and arrive at that, you know, and then that would drive the number of plex. We know that we can do 80 plus. Is that reasonable to roll out? Because that's a throughput issue. So thinking more in terms of the cell populations.
Also, if you comment on the point of view, a lot of those markers are highly correlated with each other. So often when you have a real large are together, so often I feel having high quality and a lot that are just correlated with each other is often a better case.
I'm going to have to end in a minute. They're going to drag us back to the Collins plenary. We will have a longer time span this afternoon to expand on this and to try to come to some kind of action item. So this is a good start. I hope we'll have a bit more participation and that we can solve some of the audio problems, since everybody seems like Cher trying to sing a song with a vocal modulator right now. If there are any last minute comments people want to make in the 4 minutes we have left, give that a shot. But if you think you're saying something critical, please type a brief note into chat so we can record that.
I don't know if I wanted to make a comment about the validation of antibody, especially for vendor provided antibody, you know, buying from company. I think also as a community, we should probably forcing the company to actually do some of those validations and then try to take on, like, starting from scratch, because kind of, you know, this is a service they're providing and should probably demand more validation from their side as well for when they validate antibodies. So there might be also maybe a more concerted effort on that, demanding more data from the vendors for, you know, these type of knockouts or other things that are actually very time consuming for dev labs to do in a large scale. So maybe if there are any comments, that would be also interesting.
Thank you. I think one of the issues is many of the antibodies are actually generated for flow or for uses, not actually for tissue staining, and that is a different type of validation. So there are only going to be some companies that are set up or marketing things for that purpose, but you're correct that it would be good if those that claim that they work well for tissue purposes have done some level of appropriate validation to limit the amount of work we have to do. Anyone else? We will take up as a major topic this afternoon the issue of how to organize to integrate the protein-based staining with the RNA and other types of work that are going to be rolling out or have already rolled out on the spatial area. So keep that in mind, give thought to how we could make progress in that direction as well. So the task we have for us in this part of the session is to come up with concrete suggestions, plans, future actions that can advance this topic within the context of the HCA and obviously HubMap and some of the other programs that are related to this approach. We spent a lot of the time in the first part of the session talking about antibodies and came up with a couple of action items. So maybe in order to be sure we fill out the blue sheet optimally, we can start with an action plan that follows on what we discussed there and then move on to some of the topics we didn't get enough time to cover in detail and maybe make some action items concerning those as well. For example, the data sharing, standardization across different labs and platforms and so forth. So let's begin with the antibodies. What I heard this morning, and everybody should chime in if I state this incorrectly, is that it would be valuable to generate lists of validated antibodies. They probably need to be parsed by platform. And we should incorporate not only those that have been generated by the folks that are involved in this call or in the appropriate groups that each of us are involved in, but also from other enterprises like ENCODE and so on, in order to make this maximally useful. Now, some of those, and my guess is ENCODE validated them in terms of immunoprecipitation for doing things like ChIP-seq, not necessarily for tissue work. And they may not be appropriate antibodies for tissue staining, but still having that list that's validated even in that manner can be a source of information that could be useful for adding to the tissue list where people then go through the validation. So let's see if we have an agreement that those lists parsed by platform would be valuable, A, and B, who can be involved in doing that, and then C, what can we do about having either those same individuals or a different group, an overlapping group, become involved in developing standards for validation. So let's open the discussion along those lines. It can be just you're volunteering to be a participant or suggesting other modes of action going forward. There is an option for you to raise your hand. If you click on the list of participants, and then if you do that, then I can call on you so we know who's talking. So Denis was the first one up.
So I think that would be incredibly helpful also for the H10 consortium. One thing to add is that we had a discussion there to also look into, can we find unique identifiers for the individual antibodies? Because right now we use a lot of clone numbers and there's like a long list of information. How can we really have a list that also has a unique identifier? So if you work with the same clone, that you really know this is the same clone.
That's important that we do that. Usually the clone name is correct, but if different companies have, quote, different splits of are truly identical, we don't actually know. And they're obviously, even for monoclonals, going to be batch effects, especially if they're conjugates, because each conjugation gives you a different FP ratio and can change the behavior. So we'll have to think about those annotations in terms of the level of granularity that makes sense. And this comes back to how do we create standards that allow us to interpret the data sets we generate with these antibodies. Neil?
Yeah, I think two thoughts came to mind that standards, and you're talking about the antigens or the affinity reagents themselves for standards?
I was thinking about standards that would be similar, for example, in a large consortium that was doing a lot of multiplex flow. They actually, for each an individual, put around several hundred vials away, and every time you ran a flow batch, you ran that individual sample against your panel of antibodies, so you had an internal standard for comparison where you may not have tweaked your PMTs the right way. The antibody batches might have varied a bit. And so you could essentially normalize to that standard set of cells. Now, they only last so long, but then you can bridge to a new batch of cells. Now, whether we can do something like that for tissues is an issue. Whether we can get organs that don't meet the criteria for donation, but are fresh enough that they haven't undergone a great deal of degradation. A liver can give you a lot of samples for a lot of tissue sections that could be on everybody's slides if they're staining for liver, for example. So that would become a kind of standard. So you'd know what antibodies you're using, you'd know what batch you're using, but you know what you got off of your instrument on your slide, because you'd always have a little extra section. Now that takes imaging time and reagent. And so whether people are going to be willing to or able to do that, I don't know, but that's what I mean by standards.
Yeah. Okay, great. Because I have a different take on it. Well, that's really important, and I don't have a direct solution for that. But let me provide a different slice through. And can you hear me okay this time?
Yes, very good.
Okay, I used phone audio this time. So we're generating some recombinant antibodies that are renewable in the sense that they are the right clone every time, and we can molecularly characterize them like with complete molecular composition. So that reagent, barring unfolding and other conformational effects, should be the same every time. And to push that through across imaging technologies would give us a sense of variation across the different platforms, and then we can meter that out. How reproducible is it across, say, 2 labs doing codecs, or and even characterize after they label up the antibodies with their processes. There will be variants there, and this gets to one of the things Emma dropped in as a comment about covalent tagging and things like that. I think if we tighten that up and get more precise, both about the clones and the tagging, which leverages the technology to do that, is all imprinted by biopharma. Biopharma characterizes antibodies with ADC loading, all the rest. A lot of it by top-down mass spec. That sort of mini study is something that in the next 18 months should be complete in a small scale. It could be broadened out using crock-up across different consortia. But that's a different way to approach it than the standards of the tissues that you mentioned. But I'll stop there.
To Denis's specific comment about the antibody IDs, so there is the antibody registry org that I just pasted in the chat too, that it can be used to get unique identifiers, RRIDs for antibodies that will be same for the same clone across different companies. So PubMed is, I think, starting to integrate that in our antibody registry of the consortium. So maybe that can be something everybody can use going forward.
Cinema or Elizabeth, I don't know if you want to say a little bit more about at least where we started on the HubMap side in terms of the fields we were collecting for each antibody. And then maybe to Niels's point, we can talk a little bit about what we had thought about as some collaborative efforts in this space. And I'm curious as to, for me, that there's a few challenges in what we're doing. So I'm not necessarily saying that these are the right approaches, but I think at least to put it out there, this is the approach we've taken. I think to Ron's question about the value of this and looking to see what other groups have done, to me there is, given that several groups have done this and we did a quick survey at the start of our effort, I think it's worth going back and at least understanding for the context of some of the data that's been generated by these consortia. If we ever want or ever think that there may be any kind of joint analysis, then I think it's worth the effort to understand the antibodies that were used as part of their assays. And again, sort of framing this as what's the value in doing any of this? And I think it really does come back down to the comparability of the data and understanding what exactly was labeled and how it was labeled, and kind of all those nuances of the experimental details that quite often get washed over. So I don't know, Elizabeth, do you want to talk a little bit about the fields we're collecting and get the groups, get everyone else to weigh in terms of which are maybe the most important, which less important?
Yeah, absolutely. So I posted a link to what we've collected. I'll put it here. I'm hosting a Dropbox links just so we don't edit what we've put together, but we can all look at it. So this is the second version of the antibodies we're using as part of HubMap. And so initially we went through and collected antibody names, vendors, catalog numbers, lot numbers, and then the projects, whoever was, whatever group was using these specific antibodies. And then we've gone through and added RRIDs, and then how the different antibodies are validated, what host species, clonality, all that good stuff. And then collected any vendor validation that was performed on the individual antibodies, and then provided vendor published URLs. We've then split this into different modalities and have different tabs. And so we'd be happy to integrate other information as needed. We had a discussion at a codex sprint, and so what that is just a group of us came together to talk about codex data, and we discussed curating a series of antibodies that could be, or applying for a grant to curate antibodies from multiple consortia together, where we track antibody names, the RIDs, and then validate that through multiple vendors and through what different people produce, and then account for different names that other people may be using. So they might be using the same antibody but call different things. And so that's basically a summary of where we're at in HubMap on collecting antibodies information and validation criteria.
And maybe to add to that, we also, the group came up with a template for antibody validation, where what kind of data could go in, where we actually put in the vendor data for, basic validation, and then group specific data for validation for particular applications, let's say tissue staining, or if it's barcoded with DNA or other fluorophores, etc. So we are actually, I think all groups are gathering those, basically one antibody file for each antibody that they will just then upload to create a database.
I'm actually uploading that file now. Thanks for bringing that up.
Yeah.
Just before I try to open that up, are they simply filling in the table, or are they actually uploading data they use to show the validation results?
Right now, we're just uploading data, but the intention is to upload the actual validation data itself. And so initially, we're just going through and figuring out how antibodies have been validated, what different groups constitute validated antibodies. And then we're going to make the goal, it might be a little lofty, is to define what we constitute validated antibodies and then collect subsequent data. So we're still in the middle of discussing what constitutes validated antibodies, what's the minimum requirements, how does this change if you're like conjugating antibodies yourself in-house or growing them in-house as opposed to buying them from a vendor. So those are still we're still discussing the finer details of that.
I'm going to go back and raise the point I already had made before, which is that if you do your validation on different tissue samples, and especially tissue samples from different tissues, you may wind up with a different answer. That is, it may be quite specific and very sensitive in one tissue, and then in another tissue, the background autofluorescence or other proteins that are present can give you a different answer. So I think it becomes really critical to figure out what constitutes validation across both platforms, but also tissue sources. And this is back to my comment about it would be terrific if we could put together a few standard samples that people can use from the same tissue in their hands to say their antibodies work under those conditions. So I think that should be included in the work list, the object list for trying to bring all this together.
Yeah, I would just add that that's an extremely important point that Ron raises, because you could come to the wrong conclusion that an antibody doesn't work in a tissue, but that cell might not be present. Or we've encountered, you have some fun surprises like CD56, which is a marker for NK cells, but also shown to be expressed on some neurons. So when you put it in a heavily innervated organ, you see it and it beautifully works. But so yeah, it's really important to know which tissue and have a standard of tissues that we all look at.
So for HubMap case at the moment, we each group specifies which tissue are in which, for example, fixation, also like it's FFP or cryosection for the tissue type and application they are developing. So they are providing data for the particular application they are doing, not for, doing a general validation for multiple tissues that are not directly relevant to them at this point.
So in terms of crafting the document here, while it would be wonderful if there was a representative from at least every HCA tissue consortium that participated, I don't know if we're going to wind up getting that to happen, though that should be, I guess, the first goal, so that whoever is really involved in the imaging part in each of those consortia would be part of this, so we would at least have the validation per tissue, and then that group can work on figuring out methods for cross-validation as they generate these lists, because some of the lists will be unique to the tissue. The markers are not going to be anywhere else, and others will be specific, broadly valuable in that regard. So can we begin to put together at least a first pass working group of individuals that are highly invested? We've already, several people that are directly involved in the HubMat effort speak up. We are, as Andrea is leading the work we're doing in the group, have between 100 and 200 antibodies that we think are validated by our standards, maybe not up to what we would think for the entire group here, where we're part of the Thymus group in the formal CZI seed network. Are there other people on the call who would be willing to either themselves or somebody in their group do this? And then that group can reach out to the members of all the different seed networks or HubMat networks to figure out how to get this to go across all the tissue sources.
I'd be happy to volunteer, and then, or I'm Elizabeth Neumann. I could help with HubMap efforts and then reach out to some of the other people who are working on antibodies in HubMap for us.
Emma, can I volunteer you, given how much you mentioned and about how you've been working on things like the validation this morning?
Sure. Okay.
Okay, well, I think we've got a starting group.
And can I volunteer Denis, even though he's not speaking, to link this with HCAM?
Okay, I think 5 people is enough for a first working group to begin to pull the different information pieces that exist already together, such as what we, Elizabeth just sent us, and then to think about who they need to reach out to and create the network and go from there. So that is the list and the coordination. And hopefully in the course of doing that, they will begin to discuss with people this issue of standardization. I don't, we need another working group to deal with that. That can be brought up in this context, whether we can come up with tissue samples that can be tested across different labs and platforms. And that's the other thing. If we don't have representatives that go away from the immunofluorescence to the to MIBI or mass imaging, then we need to have someone who's doing that on this group so that we cover the 2 major technologies. So we're not doing that. Is there anybody on that short list that's actually directly involved in any of the mass imaging?
Using maybe at the Broad, I can reach out to the people to ask to make sure that we are part of that.
Okay, well, Denis, if you be on that first working group, that would be very helpful, and then you can cycle somebody in to help in that regard. Okay?
Exactly. I think I'm not the perfect person for the validation of antibodies, but I will try to find the right person for that.
Maybe Zoltan, if he's interested, could be actually pretty good because he's taking care of all the antibody databases in the Sorger lab at the moment as well.
Exactly. I believe Zoltan and Sammy would be good, but I want to ask them first before I volunteer them.
Okay, well, we'll put the document together with just a placeholder for, let's call it MIT Broad for the moment, and bring it back around again when we have that worked out, okay?
Hi, this is Jeff Spraggins. Can I make one point? I think, obviously identifying a tissue to share and compare performance is critical. But one thing that is probably obvious, but just to make sure it ends up on our action list, is to just define kind of the minimal criteria and methods that we're going to use to validate. I think we've found that more difficult than we expected within HubMap, just to get people to commit to here are the minimum standards. And I know it would help us a lot if we can, probably standardize that across all the consortia. What are the minimal standards for validation? So I think that's maybe the first step in this process.
So Jeff, I think that's obviously very important. I wonder if that won't flow from the people bringing together what already exists, because they'll, perforce in doing that, begin to see what people are saying is validation in their particular communities. And then as they see that there are either differences or gaps, figure out a next step strategy to work on that. And that may require tagging to other people to pick up the work effort, or they may be able to do it within a group. But it's so intimately tied to making the list that I don't know that we should split it off as a separate action at this point. But I'm happy to get input on that suggestion.
So let me just second what Jeff said. I think there's a number of things compared to when we started this effort in HubMap that haven't been as easy as we thought they ought to have been. So, even collecting information in a standardized way in terms of even just the antibodies that are being used. And you see from the table in its current format that, there are differences in the way people have entered information even on the table, let alone how they would want to move forward with any kind of shared experiments. So, I would treat it as a separate item worthy of discussion, because it would require more resources for people to buy into and to agree on. And, there's a certain level of effort where people won't actively engage as much. And, it may require a smaller subset of particularly active groups to participate in that effort rather than everybody in general. And I think there's the coordination aspect of building the list. And then I think there's some of these efforts, for example, testing in one particular tissue type that may be best done initially in the smaller group. But, again, this is just my thinking on this.
I don't agree. I don't disagree that it's going to be enough of an effort that it probably is not going to be exactly the same individual doing the same task. The question is whether to have it cascade from trying to create the list, where they see, what efforts have been made, who's already trying to do or claim to do validation, or whether we need to set up a group right now, because I'm not sure we have the full representation we need to grab the right people who might actually be doing that. For example, I don't know that there's any Stanford person here today. Gary's not on, Mike Angelo's not on, Sean, side, the people who really been thinking at level, and also codex kinds of things. So I just wonder whether we need to put an asterisk there that has to be done, but not say that we've actually come up with a working group yet to do it. But the group putting this together is going to see who's already involved in those efforts, who's starting to talk about standardization, and then they could reach out in that manner. So does that make some sense as a plan for now? I think if you have a better way to do it in real time now, I'm happy to do it, but I'm not seeing it right now.
I think just to comment, part of what we struggled with is at some level there's ideally what you would like to do. And I think maybe this is getting back to Jeff's comment about kind of the minimum agreeable approach. And then, how do you recognize there are differences across different labs? And, that at least, again, is something we have struggled with as a group, in that, we do have all of these variants on antibody labeled imaging techniques. And part of it is, can we bring everyone close enough together that we can agree on a particular tissue type to work on? And that's been challenging because, everyone's got their own favorite tissue types. They're aware of how their antibodies perform in those tissue types. There are a bunch of challenges in that direction. And there's also kind of what our ideal set of agreed on antibodies might look like for everybody to work with versus, again, everyone's historical preferences. So it's, again, I'm not disagreeing with what you're suggesting, Ron. I think it's caused much more work and it's been more time consuming than we initially planned for. So if there is any opportunity to start some of those discussions earlier on, I think it can try to help normalize or identify a smaller subset of people to start working on this.
All right, well, how about if I nominate myself for some work, which is that I know a number of the folks who've shared what they think are curated lists with me across different platforms. I'm in close contact with Sarah and are in her group who's trying to get up this at the Sanger, obviously deal with the people at Broad and at Stanford. So I could begin to put an initial group together. It needs to have more parts to it than what I just named. And then I have a coordination with through Andrea, who I've already sort of nominated to be part of this listing category, who's also involved in the validation. So we can cross over back and forth if that makes some sense. And then if there are other volunteers on the line right now, we can make an initial working group.
Yeah. Hi, Ron. It sounds like a great plan, and I'd like to nominate Jeannie Camarillo from the HubMap. And there's a committee formed, a similar-minded committee, very similar, and it's got AJ Pillai and Julie Kim, and I wanted to just coordinate that, Elizabeth, that is the committee you're talking about.
We actually have two, which has been a little confusing. So we found out as a codex consortia, or like a codex group, that so there's two. So I've actually been involved with both pretty heavily. So we found out that Jeff Spinwright and Richard and everyone who said, like, we've had a hard time really like nailing down what we're going to do in testing it amongst the larger consortia. And so what we did as a codex group is got the three groups together and made plans for a pilot on a spleen. And we actually have a plan moving forward that I just got an email about. And so what we were going to try to do is do it on a smaller scale to then bring it to the larger group to show, hey, on a small scale, this seems to have worked in this way on this one target organ. Here's what we found that worked well. Here's what we found did not work well, to try to get people to get more excited and about it. So there's two groups, and I did a poor job differentiating them, but there's two groups.
That's okay. That's good. Yeah, I don't want to make it a HubMap focused call, but that's good. It's good to know that there's a codex group that's dealing with those issues. If you would like to shoot your top one or two targets that you're using across the spleen, we could, offer what we do at Northwestern that I think you're aware of. But back to the more general point about your tissues, Ron, you're really focusing a lot on the tissue, and I think that's really important. And so we do a lot of IPMS validation, and we're going to be doing it, target specific and tissue specific. Now, there is a mix. So Snyder's lab at HubMap created a mix of tissues as a standard for proteomics, ensemble, bulk proteomics. And that could allow some things to progress along the lines that you were talking about, Ron. But in any given IPMS, you can expect 10,000-fold enrichment of your target, and that's about it in a single step. So if you mix, you're shrinking your range that you can access targets, and so doing it on a tissue-specific basis or organ-specific basis, I think that's useful to hold that line there, particularly as you access targets that are below 20,000 copies per cell.
Well, we heard this morning about the use of protein arrays. And so we really have the full repertoire, not necessarily the splice variant of human proteins on those arrays. That's not a bad, first starting point, even though that doesn't reflect the vagaries of trying to detect those same targets in a tissue. So that is one step. But I think we really should put some effort into looking at whether there are, for at least the larger organs, where you can get quite a few sections that are pretty representative from major regions, not obviously the smaller regions, you know, one part of it, of the end of one valve, you know, in the heart, to be included in the standards. And I think we should be pushing that, because then those can be sent out to multiple groups using their methods with presumably the same antibodies, even if they're the same clones, but one is a chelate, one is a fluorescent conjugate, one is an oligoconjugate, and ask, do we get the same answers on those sections? Because if we don't get those, then we really have a big problem, and we have to figure that out going forward.
Yeah. Can I just continue on this riff? And I got a question for Richard in a second. But the cell lines, like, I'm aware of like a lot of IPMS validation efforts, and they're often in cell lines. But they have done 8 different cell lines, and that mitigates some of the variants that you might expect across tissues. But using primary tissue, you know, we are limited in that. So my question for Richard is there anything from GTEx or for, you know, or the tissue availability question within HubMap with a common IRB should be solved, I think, or should solve some of these issues, but doing it across consortia might be more difficult.
Yeah, so it's not to say too much in advance, but you know, this is one of the discussions we've been having with Chan Zuckerberg folks about having more tissue types commonly available across all consortiums. So it's something we've talked a bit about, and we've sort of explored a number of ways of trying to make that happen. So I think that's one part to your answer. I mean, to me, it seems like there's, back a little bit to the validation question, it seems like there's different kinds of validation we want to do. You know, some of what Ron was saying, you know, I think there's consistency across regions within an organ, across different parts of the body. You know, there's questions about, for example, operator variability. You know, that comes up. You know, several studies we did as part of the LINKS program shows that, you know, that plays an important variable in terms of differences in results downstream. The conjugation question is important. So it feels like there's lots of different aspects where we need to at least get a handle on how do they contribute to any variance in results we see. And, you know, it sounds, at least in my mind, it's a slightly different experiment we would want to do each time to test each part of this. And I don't know if there is a way of combining multiple questions together into one kind of experimental assay to try to resolve all of these sources of potential variation. So, you know, at least that's how I think about this. And, you know, definitely we can work on the consistency of the tissue. You know, likewise, we can think about, particularly for common antibodies, distributing those across the number of labs, see how inter-lab variability varies. You know, then taking unconjugated antibodies, distributing those, saying everybody conjugate this the way you would normally do. And, you know, in the case of just for fluorophores, because, you know, nominally then the conjugation is roughly the same, but as Ron said, the loading can change, exactly how people do it in practice varies. So, you know, it sort of feels like there's many different things that we might want to validate. And part of what I'm curious about is which of these are the most important, at least this group think are the most important to try to tackle, or how do we prioritize these? Plus, you know, what's not exactly the simplest assay, but what's the most practical way of trying to get at some of these questions?
Any comments to Richard's comments before
There's an effort, and I'm not sure how widespread the knowledge about this is. It's a trans network project that's happening at the H10. So what means there was one block of tissue that was cut and then sent to different centers. And there's also a list of common antibodies, but this is very, very small and very preliminary. And then the idea was really to have the consecutive sections actually analyzed by different centers. And that would already answer questions about like, how do different centers do the processing, which antibodies do they use? And I think this can be probably for the next step done in a more rigorous manner to really like even lock down the antibodies that we are using and the protocols. But currently this seems like you're sending consecutive sections, and then we will do the data analysis based on all the different technologies and really put them side by side to show the differences.
Right. Well, that's exactly what I've been talking about in terms of having this standard for the tissue on anybody's images, but also just in terms of the standardization. And that's how all the flow was done with this one free data set. And it was very clear that the two largest variables in getting coherent data were, one, which machine you ran it on, and two, who did the gating. And we haven't talked much about data analysis. We've been talking about generating data, but not analyzing data. Now there's a separate breakout. They separated generating the data from analyzing it, which I have to say is illogical to me, but is the way things have been set out. But we can take our prerogative to include that here. So that's one topic I want to get to. And the last one is also integrating it with the other methods, because that's also how things were broken out. So there are people doing the spatial profiling at the RNA level that's separate from doing this antibody, and it makes sort of no sense in the longer run not to have those integrated. So I think we have a kind of action item on the first level for compiling lists of antibodies that are, in quotes, validated, but maybe not, and then beginning to put a different or overlapping consortium together to work on this issue of validation, which may include having to obtain these tissues, send out samples, and compare results on different platforms, which I think is going to be critical. So I want to do a short segue to this issue of platforms. There are two different thoughts that have been going around for quite a while. A couple of centers that are experts in doing these things that process much or all the tissue, and then everybody does their own. And then within that, are there common platforms? Should there even be the only commercial platforms that are actually used that are, highly standardized, or is that too constraining? So can we start with the first topic? Is everybody going to be doing their own somewhere in their own center, or do we want more coherence by having a limited number of centers that have agreed to the standards we've been talking about in how they process things, very nailed down protocols and pipelines for generating the highest quality data, even if those individual centers are not part of all the different consortia?
So Ron, I'll make a couple comments real quickly. Your points are 100% on track. The NCI CIMACs have been going through this. And what we discovered initially was we really needed everybody to try and use the same platform to develop some level of high-level concordance. And as you get to that point, you get one or two groups, then starting to add groups into the system is quite a bit easier. But if everybody comes in with their own individualized platforms and their own individualized antibodies, the noise is too hard. Start with something very defined, very locked down with a limited number, and then grow out one by one by one.
Steve, how do we figure out what those strategies are? What's the competition for doing that? Is it a commercial competition or is it a scientific competition?
So the structure they used was a scientific competition. I will say that once you start crossing certain technology boundaries, it really becomes very challenging. And it's not that different technologies are superior or inferior, but as mentioned earlier in one of the sessions, the platforms give different information, and as noted, the high multiplex and the high concordance of markers sometimes leaves you in this dark space where you're sitting there and you've got multiple markers really giving you the same information, and sometimes you're better off skinning down the markers, refining who's got the concordance, and then going back and adding information as you work forward. So our approach, or the CIMAC's approach, has been to do this technology based by technology based. So, the immunofluorescence profiles go together, the conjugated ligand assays may work together, And it may be that you go exactly platform by platform by platform, but you eventually want to see two platforms at least cross-validate.
Other comments on this platform issue is I have, so let me just say before you start, Neil, one of the concerns that I had, and I mentioned very briefly earlier, is that some of the commercial platforms are a closed system in terms of the sets of antibodies that they have available for their platform. And it's not trivial to introduce new antibodies. So if you want to expand the pool, if you try to get antibodies to new things that come out of the single cell RNA-seq that look like they would be informative that you hadn't thought about, that's not trivial in a number of the commercial platforms, especially those that use products, as again, I mentioned Miltenyi, the release antibodies, they have to re-engineer the whole antibody from scratch. It's not just a matter of importing a clone into the panel in the way that they do it. And so I have some concerns about some of the commercial approaches.
Yeah, so just to pick up on what Stephen was saying, the focus for a paper that uses the endpoint that he described, getting cross-validation from two IF-related technologies could be the goal, and a paper, which takes opportunity costs to get a paper out, but that provides an incentive for those doing the work and to, to take small bites out of this problem, where you would control the prescription of the working group would be to control the use of the antibodies. And I know that people get distracted by wanting to chase the sexiest targets out there, but this is, the study design around this is kept rigorous, consortium-based, incentivized, and I think that's the way to achieve, for example, like what Elizabeth was saying with the codex, that there could just be a codex paper that the codex community says, here's the, the recommendations out of this process. But to reach for a second technology, maybe that could be done in, say, 9 to 15 months. And that at least is a publication that could be as broad, multiple consortia. And to control the antibodies that go in would be great. And I would just say, for anybody spinning up an effort like this, I think I can offer a blanket invitation to either send the conjugated antibodies to Northwestern, and we will characterize them like biopharmaceutical industry does. That's fairly straightforward for us to do. Or to do IPMS on a couple targets. If there's an effort spinning up, then we can really regulate the antibodies that go into the assays.
Thanks.
I just had a quick comment to follow up on what Neil was saying. I've been doing research into what publication or what journals would be interested in antibody validation papers, and I have a couple targets. So Nature has like a resource manuscripts thing that like takes these kind of larger multi-lab validation studies. So there's actually a lot of, and there's some more after that or beyond that. So there's actually many journals that are interested in publishing these types of studies, even on less exciting targets. So I think there's a huge incentive to do this.
So shall we make this the task of the second group we were talking about when they're talking, this is really a validation issue, is to try to not only validate the antibodies per se, but to ask, can we, with a common set of whatever we think are validated clones that people agree on as a subset, look across existing platforms within IF, but also between IF, for example, and MIBI, and see what things look like. And try to have that as a community resource of saying, we've been able to get agreement, or there are certain things that don't agree, that's basically how the techniques roll out. And then that fits with what we said about creating these lists, which would be technology specific initially, but if we understand where those cross-validations exist, then it's good to annotate those lists, gives people more flexibility. Is that a good working plan for this next step?
Ron, I think it is. I still agree with your problem that you brought up earlier, though, and that is you get closed platforms and you can't bring in new markers. And I don't have an answer for you. I've been a big critic of this in the past, and I try to be very polite about it, but some of the validation strategies for the big platforms have really locked themselves in such that it's really hard to do this.
Well, I think the platforms that we're talking about right now, with one exception, are, quote, open. And I don't know all the IP issues about bringing other antibodies into the so-called closed platform, but that's something that can get brought up in the discussions. You know, there are issues between Fluidigm and whatever the MIBI company is, whose name escapes me at the moment, in terms of cross use of the metal chelate antibodies.
Ion path.
Yeah, so there are things that will have to get worked out, but I think the group we're talking about has their fingers on sort of all these different pots. There's Codex, there's MIBI, there's our own, you know, more academic approach to doing this. There are one or two others. I think if we put that group together, we'll see who's willing to play in the same sandbox in trying to do the cross-validation, and that's what will get reported out.
Can I ask your knowledge about flow and the whole history there, you know, with all the CD markers? How did that get stood up over the years, decades? And it's both commercial companies can focus on that and buy in, as well as all the, it's an incredible story there. What can we learn from that?
That's probably longer than we have time for today, Neil.
Okay, oops, that's a lecture. All right, well, I'll go read the history. Okay.
Because as I said, even very recently, large consortia going to high density flow needed to validate reagent sets, create lyophilized antibodies, and then exchange standardized cell pellets or tubes in order to come up with something close to standardization. And as I said, the answer at the end of the day was whoever gated your data gave you different answers despite all of those standardizations. So until there's a machine learning basis that everybody agrees on for doing the gating, you still wind up with data that don't agree between even the same center on the same machine, but two different people gating the data manually. So,
but the tactics to bring in the companies, like how, I mean, we're big enough that we can actually flip that script over the years, I believe, and get them to, I don't know, go ahead.
Yeah, they may. I think if it's going to look like the whole system is going to roll out in a particular direction and they'll get left out if they don't expand their pool, they'll have to look at expanding their pool of reagents to be able to cover the waterfront that they need to cover. So that we'll see. I don't know how that's going to play out. Obviously, as an NIH person, I'm not so tied into all the commercial aspects of this.
I think one way is if a vendor is saying an antibody works for IF or IHC, it should be entirely possible to demand to see the data and for them to share the data. So we could essentially use our community power to ask for this data either to be online from the beginning or at least to be provided on demand. So like some of the assays, validation assays really will not need to be done from scratch, but can be done in an application-specific way in the labs, but at least we would have some basic data from each vendor.
I've been actually communicating with most vendors about getting their antibody validation, and some play nicer than others. So I'll say Abcam's been the nicest so far about giving me the most detailed information. And several others are saying it's proprietary and won't give it to us.
So that's what we should really resist. I mean, CST also has been really nice, and they're really helpful. If you want additional information, they can actually share the additional data they have. But we should really, resist those companies who would not share any validation information, because that's just not appropriate.
Oh, I agree, 100%.
Yeah.
All right, since we only have about 25 minutes left of the session, I was hoping we could move on to a couple of other topics that we may need to take some action on. As I said, even though the things have been broken out, we need to think a little bit about analysis and also about integration with non-protein-based imaging platforms. So if we can talk a little bit about analysis. because that also goes into any of this validation. If you're sitting there sort of just eyeballing whether you think you've got good staining or not, I'm not sure that that's going to become a extensible standard for looking at things, and especially as you go to higher and higher plex. And I know Denis and others that are on this Zoom meeting have been involved in trying to deal with some of the software tools we need to avoid what I just described for flow, which is, you know, people sitting there doing it all manually. So can we have a brief discussion of where that sits, maybe from some of the HubMap folks as well? And how do we want to try to integrate that into our validation strategies and platform selection strategies?
Profiling of, I mean, I was interested in spatial profiling and profiling.
That's where I'm going, right?
Yeah. Yeah, not many people are there.
Is there someone saying something, or is this just not muted?
I think
Someone needs to mute.
I think Bob Coffey was on, so everybody else mute. Denis, go ahead.
Sure. So I'm gonna moderate the session in the afternoon where we're gonna talk about the analysis part. I think briefly what we also trying to do with this TNP project that I mentioned, where we distribute the tissue across the centers, is also just to see how, so as you said, like everyone can gate and use different tools to do the things, but then to see what is actually robust. Like once we give the people the freedom to use different algorithms to gate what they think is the right thing, what is like robust, what we get out in the end, and what is very sensitive to those kind of tools. I think this is one of the key things that we also try to include now into algorithms, to not just say black and white, but to kind of have a probability of this cell type to be the cell type that we assume it is. And for some others, it will have very very high sensitivity to the parameters. So this is at least how a lot of people that I'm talking to address this issue of like sensitivity.
So do we need to do something of the following type, which is to have real expert manual creation where people agree on a couple of unknowns and then run equivalent to a dream challenge for protein folding or crystallography, and send those data sets out to everybody creating algorithms and say, how close do you come to what we think the ground truth is? Is that a strategy we ought to be following?
If we can get ground truth for those type of questions, that would be amazing. That would be, of course, the dream.
Is that something that the HubMap people are doing?
No, HubMap is not doing this yet. I think your vision is very, very good. As you noted, it's what we believe the ground truth is, because the ground truths here are a bit ephemeral. And I like the proposition.
I mean, I think if you get 4 or 5 people to go and pore over the manual segmentation and other types of algorithms for looking at particular sets of tissue, they'll come to some reasonable agreement of what the best expert ground truth is, which doesn't have to testing the algorithms. We'll have to leave open the possibility that some algorithms will discover things that really are there that were missed by the people doing the curation, and that'll have to be looked at. But I think this is the strategy that's worked in a number of fields when you're trying to develop algorithms where you have some way of really knowing, do they work and are they efficient? And so maybe we can begin to put after Denis, after your session, the end of the day, we can circle back and come up with a group that wants to undertake something like this. Because we've tried to go between manual curation and some of our AI algorithms, machine learning algorithms. And the answer is, yeah, we can hit 98 to 99% for certain very easy things. And then when you get to more difficult ones, it's extremely tricky to do that. And so when you're the high multiplex we're all talking about, getting robust algorithms that enable sort of non-biased analysis is going to be absolutely critical. So we definitely have to coordinate with your work, your breakout this afternoon. Any other comments on this particular topic?
So Ron, I commented that HubMap is not doing this. However, in fact, it was somewhat envisioned. What they were going to do was different groups were going to use their own internal segmentation systems. Then we were going to cross the segmentation systems across tissues. Then we were also trying to make the data sets available so that external runs could be done. The idea, complex as it is that instead of, many of the systems are trying to run multi-cell type segmentations concurrently, is rather move to a tiered cell type segmentation model where you segment out one cell population, then another cell population, and another cell population. I don't think any of your comments are contradictory to some of the views that are being pursued in HubMap. I think this discussion opportunity is really beneficial.
Well, we're supposed to not comment a lot about our own research here, but I'll just make one comment in this regard, which is that there are a number of groups who are now working on approaches that don't do any real upfront segmentation, but are based on pixel analysis that may turn out to be the most efficient approaches for doing this, given the nature of the data sets. So I think there's a lot of room for doing this. And as I say, Denis is running the session this afternoon, and I think we should circle back and have some more discussions. Neil?
Sorry, I was chewing.
Okay. So let's spend the last couple of minutes on one or two other issues. So one of the things that never really seems to come up, because it's already hard enough to think about doing this in sections, is whether we need to be doing things in volumes. We're not, we never talked about doing serial sections for reconstruction. And so if you're trying to understand, really creating an atlas in a tissue is 3D, how do we do that? And so, or should there at least be a working group that's trying to solve the problem of moving from multiplex in sections to multiplex in 3D?
Yes, there must be.
I mean, to be honest with you, pretty much all the same questions and standards we talked about apply here. It's just a bit more complicated because you're adding another dimension that also is not an ideal dimension for the way typical microscopy is done, although there are solutions to that to get to isotropic resolution. So this is not immediate, but it is something that I think we need to do, where we've been working on it with Gary for a while. It's not completely solved yet to go to very high plex, but we think there are solutions that are pretty close. And so that's something the group ought to think about. And so maybe we can spend the last 10 minutes on something that's, again, one of the sub-breakouts today, which is where are people both practically and in their thinking, and what do we need to do to integrate between the protein work and the RNA work in terms of getting everything done in a one-to-one spatial mapping, which I think is the goal most of us have. Anybody have a working method for doing high-plex RNA and protein at the same time?
It depends what you mean by high-plex. Hi, Zoltan here. So I don't know if anybody has worked with nanostring yet. So they have usually a 4 to 5-plex protein, followed by a pretty broad, 1,000+ plex spatial transcriptomics, where it's not single cell resolution, but it's regional. And then we're developing a method as well where we have picking from regions, and then the adjacent section, you can then do multiplex imaging. But on the same section, it's quite challenging.
So I think that's exactly the right points, and we ought to say that we need to set up a working group. And again, we'll have to figure out, if we don't get all the volunteers online, we'll do it as a follow-on for where people are. We were working until we got shut down with Sonia on the Illumina array system, integrating it with somewhere between 15 and 20 multiplex on the same section, but I don't know where that's going to go while we're shut down. And so there are a number of things out there. Same thing up to 16 to plus plex with riboprobes in ACD now, but where we had to back off on the harsh conditions in terms of doing staining at the same time, but it looks like that may work. So there are a couple of things, none of which are nailed down yet, but I think we ought to, again, circle back to the subgroups, so Xiaowei and the other people, and see what they came up with today, and then try to coordinate another action group with them to combine the antibody parts with the oligo parts.
And I've seen one manuscript that's going about 5 plex on both sides. That's as deep as I've seen more robust data at this time. You know, I think that this is like the 3D. It's a tough hill.
Yeah. Emma, you had your hand raised.
Yes, I wanted to ask if someone tried to combine codecs with in situ sequencing. We were just about to start trying to combine those two protocols when labs shut down, so we haven't gotten any data yet, but I'm out of curiosity.
I only heard the second part, so start over again.
Okay. I was wondering if someone here tried to combine in situ sequencing and codecs. We were about to start that, trying to combine them back to back on the same section, but we haven't gotten any data yet.
We're doing some work combining it multiplex with mirror fish, but again, very early days and not into sequencing of the more fizzy kind of side. So I agree, it's challenging. We had a method working where we could do some multiplex and a couple of RNA probes, but we didn't think it was robust enough to roll out. So we backed off and started again with a couple of different approaches. So I think we're all in about the same place right now. You can get a couple of this and a couple of that working, but doing a lot on both sides routinely doesn't seem to be quite there yet in terms of anything I'm aware of, which doesn't mean nobody's done it. So I think that should be also, though, on our action list. And if any of the people in the group want to volunteer for being conduits to the groups doing that, to say we're working with the Broad group as well as ACD as a company directly on that. So we've got a toe in that, but there are other people obviously put a lot more effort. So if you can send me a note so that I can update the blue part of our form, then we can have another action item that would go there as well. Are there any other topics people think we need to cover before we close? We've got about 9 minutes left, technically. Everybody looks pretty ready for a break.
If I might. Yeah, I'll just ask very briefly. So it's either you have low plex protein and high plex transcriptome or vice versa. Is that just a trade-off because the detection modalities are different, and then specifically that if you want to detect RNA, that requires limited proteolysis, and that some protein epitopes are more susceptible to proteolysis than others?
That seems to be the limitations right now. I'm not convinced as we evolve the protocols that it will remain the limitation. I think people are having to learn how to sequence these experiments correctly, and we're so early that they have not explored all of the options with reference to buffers and molecules that can be placed in buffers that may have differential protective effects at times.
Yeah, so just to go back again, yes, it is having to remove the protein to get good RNA sequencing, at least quantitative work, that is a problem for the staining. One approach several groups are taking right now is to go back to the first step of expansion microscopy, and something that's being done also for just merfish as itself, and actually do the staining first in fixed material, and then cross-link the antibodies into a matrix that's not proteinaceous, and then dissolve everything away in order to make the RNA accessible, and then do your RNA work. And that gets around this issue, but then you have all the chemistry that Steve was just talking about. How do you make the matrix? How do you cross-link things in? Which oligos can you use? How do you be sure that you don't have probe overlap between anything you use for your antibodies versus the RNA probes? And so there are a lot of little details that have to get worked out, but in principle, there are methods that can work and that wouldn't be as difficult as in situ sequencing over extremely long periods of time. So that's my understanding of where things are at the moment.
And in general, for tissues, protein and RNA detection protocols have been not very much optimized to get the best of two worlds. So there is definitely a lot of room for protocols in terms of sample preparation and probe labeling, etc. We're also trying to develop in situ amplification with multiplexing for RNA and protein on the same sample, but we are still at a very early stage right now.
Well, that's why I say I think this ought to be our end action item to circle back to Evan and Joe's sessions, see where they wound up, and then maybe put an overlapping group together to really see whether we can be exchanging information well before publication dates, where you have to go back and revisit things, but really working and exchanging information to try to optimize these as soon as possible. Because each group has some little tricks, but I don't think we've put the package together yet anywhere. All right, that's my message, that we have 5 minutes left in our countdown to the end of the session. Anything we need to circle back to do or to discuss?
So one quick question. Are there any existing groups or meetings or working groups or other activities that we may want to try to take advantage of here? You know, it seems like we're proposing setting up a lot of new things. And there's always a barrier to doing that. Is there any way, or is anyone aware of any existing groups that we also may want to tap into?
Richard, I'm going to give you an interesting answer, and that is there's been a lot of conversations, but the groups haven't been large enough. I think this group, because of its cross-pollination and size, may have enough energy to overcome the entropy of these discussions. It's really been an issue where we just haven't had enough people at the table with enough data. Whether it's the RNA protein or it's the 3D or even some elements of the validation. I mean, I know of manuscripts and discussions that have gone on, but they stall out.
I mean, Richard, I'll point out that at the Toronto CZI seed meeting, there was what looked like a consensus about how to move forward with some tests of multiplex work for groups that had not really come up with a method that would work on their tissue and where they could try to cross it over to their RNA data, at least based on key molecules. And that was a breakout session like this. It was about a quarter of the total to the big session and opened it up to the floor where there was no real discussion of what happened in the breakout. And of course, nobody else voted for it. So it looked like a pretty sparse vote. And CZI said, I'm not going to really go ahead with it, even though it was almost no cost to them to do it. So there is this cycling that keeps going on. The best that I can say is that we are working with both Sarah and Aviv on these issues. So at a certain level, we sort of are touching the people who should know about what else is going on. And at least they haven't told me that they know anybody who solved any of the things we're talking about when we talk to them about it. But I haven't been able to get an update. Obviously, I believe was very busy in doing all the COVID and this other stuff, and Sarah as well. So we'll have to cycle back and see where things stand. But I was up at Broad right before all this happened, talking to Aviv, who, in her opinion, didn't see that many of these multiplex systems had been validated out of their originator's hands. And that's obviously part of the validation story that we discussed in the first part of the meeting.
Ron, I want to emphasize that there seems to be an issue of people think, oh, the first validation's enough, and that's not the case. And it's people are strapped for sufficient funds sometimes to carry on more robust validations. We've run into that challenge with one of the bigger projects where it's been like pulling teeth, and I'll turn around and I'm not getting money for this and it's not my systems, but sometimes the funders are putting the money on them too tight to let them do the validations they need to in these systems.
All right, everybody, thank you. I think we actually got to the goal. Hopefully the scribes have filled out the blue section, or we'll be able to come back and do that with a couple of action items, a few names in the first group, maybe in the second group, and then some thoughts about what we ought to do for the third and the fourth ideas that we talked about today. So thanks to everybody, lunchtime, and we'll see you back for the afternoon session.